DNA contains a genetic code that living cells can read, understand, and express. DNA is made of just 4 nucleotides joined together in double-stranded molecules that can be millions of bases in length. What exactly do those bases code for, and how does the cell read that code? That's where RNA comes in. RNA helps to put the genetic code into action. RNA, like DNA, is a nucleic acid that consists of a long chain of nucleotides. Genes contain coded DNA instructions that tell cells how to build proteins. The first step in decoding these genetic instructions is to copy part of the base sequence from DNA into RNA. RNA then uses these instructions to direct the production of proteins, which help to determine an organism's characteristics. Like DNA, RNA is made up of nucleotides. Each nucleotide consists of a 5-carbon sugar, a phosphate group, and a nitrogen base. However, DNA and RNA differ in 3 important ways. RNA uses the sugar ribose instead of deoxyribose. RNA generally is single-stranded, and RNA contains uracil in place of thymine. These chemical differences make it easy for enzymes in the cell to tell DNA and RNA apart. The differences between DNA and RNA allow them to perform separate functions in the cell. The information in DNA is always around, stored safely in the cell's nucleus, where it serves as a template to make multiple RNA copies. In contrast, RNA is synthesized when the products of a particular gene are needed. RNA copies travel to the ribosome, which then put the coded instructions into action by assembling proteins in the cytoplasm. Some important vocabulary for this section. Messenger RNA is a type of RNA that carries copies of instructions for the assembly of amino acids into proteins from DNA to the rest of the cell. Ribosomal RNA is a type of RNA that combines with proteins to form ribosomes. Transfer RNA is a type of RNA that carries each amino acid to a ribosome during protein synthesis. Transcription is a synthesis of an RNA molecule from a DNA template. RNA polymerase enzyme links together the growing chain of RNA nucleotides during transcription using a DNA strand as a template. Promoter is a specific region of a gene where RNA polymerase can bind and begin transcription. Intron, a sequence of DNA that is not involved in the coding for a protein. Exon, express sequence of DNA, codes for a protein. RNA has many roles, one of which is protein synthesis. RNA controls the assembly of amino acids into proteins. There are 3 main types of RNA involved in protein synthesis: messenger RNA, ribosomal RNA, and transfer RNA. Each type of RNA molecule specializes in a different aspect of the job. Most genes encode instructions for assembling amino acids into proteins. The molecules of RNA that carry copies of these instructions from the nucleus to the ribosomes in the cytoplasm are known as messenger RNA. Proteins are assembled on ribosomes, which are small organelles composed of 2 subunits. The subunits are made of several ribosomal RNA molecules and as many as 80 different proteins. During the assembly of a protein, a 3rd type of RNA molecule known as transfer RNA carries amino acids to the ribosome and matches them to the coded mRNA message. A single DNA molecule may contain hundreds or even thousands of genes. However, only those genes being expressed are copied into RNA at any given time. The process of copying a base sequence from DNA to RNA is known as transcription. In transcription, segments of DNA serve as templates to produce complementary RNA molecules. Transcription is carried out by an enzyme called RNA polymerase. RNA polymerase first binds to DNA and separates the DNA strands. It then uses one strand of DNA as a template to assemble nucleotides into a complementary strand of RNA. A single gene can produce hundreds or even thousands of RNA molecules. RNA polymerase does not bind to DNA just anywhere. The enzyme binds only to promoters, which are regions of DNA with specific base sequences that can bind to RNA polymerase. Other regions of DNA cause transcription to stop when an RNA molecule is complete. New RNA molecules sometimes require editing before they are ready to be read. These pre-mRNA molecules have pieces cut out of them before they can go into action. The portions that are cut out and discarded are called introns. The remaining pieces, known as exons, are then spliced back together to form the final mRNA.